So our last speaker of the session is Matt Benning. He received his degree at the University of North Illinois, and then he went to work in Hazel Holmes' lab at the University of Wisconsin-Madison. Stayed in Madison and has been working for Bruker for a number of years now. And you told me you're mostly a small molecule crystallographer.
I was a small molecule crystallographer. Now
molecule.
I'm a protein crystallographer.
I like that. So, Matt, you're all set.
Okay.
He's got it. That microphone does all the recording.
Oh, okay.
Okay. Thank you.
Thanks, Jim. So today I'm just going to briefly show you some of the protein software suite that we've developed at Bruker over the last decade. And it's sort of an offshoot of small molecule software that Bruker's been developing since the 90s. And so now it's been optimized for macromolecular work, but carries along some of the features that small molecule crystallographers have been using for a long time when dealing with very problematic samples like twin to cracked crystals, weak diffractors, etc. So what I'm going to just show you today is some examples of a couple of the features or tools, one primarily called RLATT, which is a really nice tool. It's a reciprocal lattice view. And so what it allows you to do is this is a simple program that allows you to look at the reflection arrays in reciprocal space. And it's a very good way of getting an idea of what's going on with your diffraction. And I'll show you some examples of using that for both twins and for just a sample that was not very good. So NOW is a program written by George Sheldrick, and we've been partnering with George Sheldrick for a while now. He writes some specific programs for us for the Proteome and our small molecule suite Apex. And there's a program called NOW that helps to automatically detwin crystals. So we'll find the different domains, orientations of the domains in a crystal, and give you the twin law between them. We have an integration software along with the suite. So Proteome is a full data reduction suite. And SANE is very similar in a lot of ways to XDS as far as how it approaches integration. It does a three-dimensional profile fitting, which is very useful for twin crystals, for example. And then a program written by George called TWINABS, which is for scaling, doing absorption correction and scaling twin data specifically. So a twin is just an aggregate of a crystal, two same crystals with a specific orientation. So you have two crystals, they're rotated from one another in either a specific way, like 180 degrees, or an arbitrary. And so you need to find out what are the orientations of each domain and the relationship between them. And there's different types of twinning. So the first type is merohedral twinning, in which you have a twofold that correlates with the metric symmetry of a crystal system, but not with the Lowry symmetry. So, for instance, this can be possible for higher symmetry space groups like tetragonal, trigonal, cubic. So you might have a crystal that indexes and looks like it's P4 upon M, when actually it's P4 upon M, with a twofold relationship between two different domains. And that relationship we call the twin law. So it's mimicking higher symmetry because there's two different orientations of essentially the unit cell and the crystal. In this case, the two diffraction patterns overlap completely. So just looking at the diffraction pattern, you would think nothing was wrong. But you don't find out later until you either look at the intensity statistics, as has been mentioned today, looking for effects of twinning. And one of the statistics that George uses a lot is normalized structure vector values, or statistics based on E² minus 1 values, they're called. And they're just looking at the distribution of intensities. So for centric or acentric crystals, the intensities have a similar distribution. For centric, you find a value around 0.97. For acentric, around 0.74. And these are the same for all crystalliforms. So when the intensity statistics deviate from that, that's a sign that there might be something going on. So in this case, for merohedral, one of the advantages is you can kind of treat it as a regular crystal and just treat the structure, solution, refinement in the same way. So a lot of the programs now will take, if you can find the twin law between the orientation between the domains, they'll take that information and refine against and find the twin fraction, how much of each domain is present in the overall aggregate. And so there's some problems when you get to what's called perfect twinning, which is 50-50 split between the domains. But for the most part, it's a little easier to deal with. There are other types of twinning that are a little more difficult because they're not just straight overlaps of the reflections. So in pseudo-merohedral twinning, metric symmetry doesn't constrain the patterns to overlap directly. So you can have partial overlaps, you can have stronger overlaps. And in this case, the twin law is in a little higher crystal system than the actual Lowry symmetry. So for instance, if you had a monoclinic cell that had a beta angle close to 90, that would mimic orthorhombic, and you could have a twin, pseudo-merohedral twin. Another case would be an orthorhombic crystal with A and B, or two axes very similar to one another, which would mimic a tetragonal crystal system. So the crystal system of the twin is higher than the actual Lowry symmetry. And so you can see in the pattern now, you start to see some patterns in the diffraction that aren't really normal for a single crystal. One interesting one is it's called a partial merohedral twin. And this is kind of interesting because in this case you have complete overlaps and no overlaps between the spots. And they'll happen in rows or layers. So if you're looking at your spots, you see that the red here, those are complete overlaps, and then the next layer over are non-overlaps between the spots. And that something like this would have what we call an index of 2. And so it's kind of interesting that you get this pattern of every other layer, complete overlaps and no overlaps. And then finally, there's something called non-merohedral twin, which probably a lot of you have seen, but really haven't dealt with. And everyone has crystals that, maybe not grow single, or in some cases you can kind of call them cracked crystals, non-merohedral twins, but we get kind of strange between calling what's just multiple crystals and what's a really true twin. But in this case, the twin law between the domains is arbitrary. So the crystals can be oriented in sort of any way, but a lot of times it's similar to other twinning where it's 180 degrees between the domains. You can tell in this case where you see that it almost looks like you do have intersecting lattices. So in the non-merohedral, you can really pick up in the patterns something strange is going on. And you can see typically you have them usually along cell axis, or a lot of the peaks will be split, where other peaks will be nice, show nice profiles. So it really does pop out in the diffraction pattern. So the program I was talking about, RLATT, is just a reciprocal lattice viewer, and it's a simple program, but it actually is very good at helping sort of figure out what's going on. So here is just looking at the reflections we've harvested, and it just shows you in a simple space what those reflections are. And we were talking earlier about indexing and different types of indexing techniques, and this is sort of a one-dimensional. Jim was talking about doing a histogram in one dimension. And at the very top, you see a histogram of the spots, and that as you get peaks up there or spikes, you can see that that's just an indication that in that direction there's a high density of reflections. So indicating that could be a unit cell vector. So one way that RLATT helps you is you just look at what the reflection array looks like. And when I can index something, the first thing I do is come in here, and it's usually quite obvious what's going on. It's got a bunch of graphical tools that actually, it was originally written as a teaching program, so it actually has a lot of ways to display unit cells. But the thing that's most useful is just editing and visualizing the reflection array. And I can rotate this in 3 dimensions, I can add different scans, and it'll all show up exactly how it would be in reciprocal space. So this is an example of a penetrant twin. It's bovine insulin. This is grown in George Sheldrick's lab. And in this case, the crystals grow from one center but grow out as multiple crystals. And so if you look at the diffraction pattern, something doesn't look right, right? You see a lot of spots that are very close to each other that just don't really fit the general pattern. So looking at this, you would throw some red flags, spots like here and here that just don't make a lot of sense. So if you look at it in RLATT, it really pops out that there's just sort of a strange distribution of spots in the reciprocal space. As I rotate it 190 degrees, then it's really easy to see the two overlapping lattices in there. And what I can do with our lattice, there's a number of different editing tools. I can edit a box, I can edit a circle, take out an individual spot if I want to. But the most useful one is the lattice line. So I just draw a series of lattice lines out, and I can line up the lattice lines with the lattice that I'm trying to isolate. And by doing that, I can then assign it to a different group. So now I've separated the two lattices. I've got the red, which is the reflections from one domain, and I've got the yellow, which are the reflections from the other domain. And now it's really straightforward just to separate out the other domain, and now I've got one reflection array. So just by doing that, simply, if I go back to the integration, our indexing software, is now I have two different reflection arrays. So essentially, I've just de-twinned the reflection array. And now I can take that and index both of those and have two different orientation matrices, and then I've essentially de-twinned and indexed. So here you can see the overlays for both the cell that I've indexed from the first, the red array, and then yellow. And you can see for a non-merohedral twin, you'll see a lot of what we call singles, which means there's no overlap between the two patterns. And you also see some composites where there's overlap between the two patterns to different extents. So that's what you're trying to deal with when you have a non-merohedral twin. So this is an interesting case that I did. I got some crystals from Rebecca Page at Brown University, and it was a protein phosphatase 1 complex of DNA, and they were having trouble solving the structure. So we collected a data set, and it easily indexed in an orthorhombic F cell. And here's the unit cell that popped up. And the index was great. It matched all the patterns, matched all the spots, integrated the data. Our merge was 20%, and it's throwing out 20% of the data during scaling. So something's wrong. So if you look at the Brave search, there's another cell, which was C2 with 130, 54, and 140, with a beta angle of 117, which looked fine, actually integrated, and scaled well. But looking at the intensity statistics, E² -1 value, you got a value of 0.94, indicating a centrosymmetric intensity distribution. And also from the systematic absence, there looked to be a sequelae. Both of those are not consistent with protein crystals. So that's a big red flag. So what's going on? So going in RLATT and looking in one direction, we see a zone, and everything looks fine. The spots look well separated, the lattices look great. Turn it 90 degrees, and you start to see something strange. You see that every other row, you have a single layer of spots. And then the other row, you have two spots that are very close together. And this is what we saw in the introduction, it's a partial merohedral twin. These spots are overlapping every other row, and then the row between those are the individual spots in the individual domains. So at RLATT, I can just simply take one domain, highlight it, change it to a different color, and then take out the other domain. And now I essentially de-twin. But in this case, it's a little different because you've got complete overlaps in the layers. So looking down H star, what you see is that here are the rows, the two domains together. You've got the red and yellow, where they're separate, the singles, and then in between you have white where they're completely overlapping. And so the true cell looks to be C2, but it looks like instead of 130, 54, and 140, it's 130, 54, and 70. And you see why the indexing is thinking 140, because it's seeing the two singlets right close to one another. And so now, essentially, all I have to do is create the two different arrays for the two domains. You can see that one is shifted with respect to the other, as we would expect, and then go back and index, and that's how you can separate the twin. And so here's the red domain. You can see it lines up on different spots than actually the yellow domain, but there's still a lot of overlaps because every other row is completely overlapping. So you see here you've got red and yellow, you've got a number of singles, but a number of overlaps. And so that's how you go to the next step. So because I've already indexed and have the orientation matrix for both domains, I can go right into the indexing. So this is passed on to Saint. Saint does a three-dimensional model profile in a similar way that XDS does. And so it's important, I think, for twins that you use the three-dimensional approach because you can actually build model profiles for each of the spots. So the three-dimensional approach works where you take reflections of certain signal and noise in a region on the detector or the overhaul detector and build a model profile of what the peak looks like. And then it applies that over to a position. So what Saint does, for the spots that are singles, it just does the integration as normal. But for the spots that are overlapping, because it knows the profiles of the peaks and knows the positions of where the peaks are, it knows those peaks are overlapping, and we'll try to deconvolute the composite reflections. And so here what you see is this is sort of a partial overlap, where you've got the blue is actually one spot, and then the red is another spot, and then the black is what it actually looks like on the detector, or the aggregate. And so that's what you're trying to separate. So what Saint does is it produces single reflections for those that are non-composite, and then for the composite reflections, it'll sort of partition, come up with an intensity estimate of each domain contribution, and then an aggregate sum. And it passes that on for scaling. So the next program would be TWINABS, which is how you would scale the data. So TWINABS is a sister program to StatAbs, a scaling program George wrote that does an absorption correction based on the approach that Bob Blessing did back in the 90s, where in this case he does a multi-scan approach. So he looks at the same reflection in different orientations of the crystal and tries to figure out what the absorption of the crystal based on that is. And so he models it on spherical harmonics. So he applies an absorption correction, does the fitting, and then tries to detwin the data. So enabled to calculate what the scale factors are for each one of the domains. There's a couple ways of doing it. First of all, you can calculate the scale factors just based on the single. So none of the composite reflections. If you have enough singles, that's fine. If you have enough data, you can get the scale factors and apply that to the composite reflections. If you don't, you could say have a dominant domain, so one could be a lot stronger than the other crystal domain. So then you can take singles and composites from that dominant domain and apply that to the second domain. And then the last way is if, say, you had 4 components and you had 2 dominant ones, you could add the singles and composites, determine the parameters from the 2. So there's different ways of scaling. So the output for TWINABS is 2 different ways. He outputs D-twin data and twin data. So he has an HKLF format in which, he solves a set of almost linear equations for nodes that we have. So we have the measured intensities of the reflections. And then we look for the unknowns, which are the twin fraction, how much of each domain we have, and then the actual the contribution of each domain. And that's here. And then you can solve these using the partition information from Saint as a restraint to keep it more well behaved. And so basically you detwin the data, you get intensities matching a unique set for each domain. So what's nice about this is that you can do both structure solution and refinement from this. But to improve the refinement, he also writes out what he calls an HKLF5 format, which is just the aggregate sums of the reflections. And so if you look here, this is actually the untwinned data, and then in the file, he's grouping in the reflections that are part of that aggregate. So it tells the corresponding programs this aggregate is made up of these reflections. And so from his experiences, it seems like the HKLF4 works really well until the end, but actually refining against the twin data actually produces a little better result. So he usually solves a structure, refines it with the HKLR4, the D-twin data, and then does a final refinement against the twin data. So for the phosphatase 1, you see that this is the twin law between the domains. When I actually split the two, I got a twin fraction, one domain 64, the other 36%, and then splitting them, I get much better data. The R factors, we already talked about this all day today about how to correlate what the data looks like, but for an R merge for these, you get a relatively, what you would expect for a decent data set. And then when I did this, the E-squared, or the intensity statistics, the E-squared -1 values were normal. They were what you would expect for an asymmetry. So it really looked like C2 was the correct cell, and that it was a partial merohedral twin. You can also use RLATT for things that just don't frac well, not necessarily twin. And this is an example. This is from, you probably heard about the data processing session. And this is a sample of a trypsin with a tightly bound inhibitor. And they did got the data, and they wanted to see if they could solve the structure. And at phi 0, it looked fine. When you rotated about 70 degrees, the refraction really fell off. The spots were overlapping. You went a little farther, radiation decay set in, and the data set was pretty much drawn by the time you got to about 120 degrees. They had solved it, they thought, in P43, but it doesn't scale very well as tetragonal. And they could only see the inhibitor in the molecular replacement structure, so it was kind of strange. And then they tried C2, but they couldn't get a molecular replacement solution. The data processed okay, but no solution. So if you look in one direction, the lattice looks fine. When you go the other direction, you can see how there's all kinds of overlapping spots. So it's hard to index, and I had trouble indexing it. But as you rotate a little bit in RLATT, you can kind of see what the lattice lines are, and then you can easily start to separate all the noise that's in there. After that, it was easy to index it, and then it was just trying to figure out what was the correct space group. In this case, it was hard to tell which one, so I just integrated, as Kai talked about, P1. Looking at expert, you could see that orthorhombic shows good merging statistics, and then tetragonal, the merging statistics go way up, so it looks like it's orthorhombic. And then using the Trypsin model, I was able to get a solution in phasor. So the space group probably is P2221. And then finally, I just wanted to mention a program called NOW, which is George's brute force way of detwinning a twin crystal. And in this way, what he does is he tries to find, starts with a random set of vector lengths that you sort of specify, kind of how long the unit cell vectors are for your crystal. And then he just calculates a family of planes for that, looks for reflections that lie along those planes, and tries to come up with an index for what matches most of the crystals there. Well, once he's done that, once he's indexed one domain, then he just rotates that cell in a circular space looking for a match to the rest of the reflections. And in this case, this is the insulin I was showing earlier. For a cell now, you just put in a reflection array. It comes up with potential cells. You select the one you want. It then tells you what the orientation matrix for that cell is. And then if you tell it to go on, it'll keep looking for the next domain. And so in this case, it found domain number 2. It tells you this is what the orientation matrix is. This is the relationship between the domains. And then now you've essentially detwinned it. So it's an automatic way of just taking a reflection ray and then searching for 2 different domains. And then looking at it, RLATT, again, you can see the 2 different lattices. So you can go back, you take that data from NOW back into Protean, look at RLATT to see what the reflections look like. So I just wanted to give you a quick idea of the Protean software. A lot of you probably aren't familiar with Protean, so if you have any questions, there are a lot of tools like this to help you deal with difficult samples like this. Thank you for your attention.